Hello, could somebody confirm you can hear me?
Yeah, I can hear you, man.
Thank you, Jaden. I was able to log in sooner because I never closed my Zoom page. So I just went back a few and was able to immediately get into the lab. Let me see where we are. There we go. Oh, lost it. Come on. Of course, you're not seeing anything, so you don't know what I'm doing, but I'm trying to call up the schedule here. Are there any questions before we begin?
I have one question. Last week you had mentioned the infectious disease project. Are we supposed to email you our diseases starting today?
I was supposed to talk about that in the lab, but we haven't started the lab yet, so that's why I haven't talked about it yet. But yes, starting today, I will be taking infectious disease research requests, project requests. So the first person who sent me an email today, and it could be anytime today, I will take that request. If you sent it to me before today, I'm not going to honor it. It has to start today because the project is starting today. You need to clear your infectious disease research project with me so that I can make sure that there's only one person taking each disease, and we can break it down a little bit. Like if more than one person wanted to do the flu, well, I can break it down into flu type A, flu type B, and what is it, influenza Z. There's actually sorry, there's actually 3 types of the flu, so I can break it down that way. I can break down a few other diseases, but it has to be an infectious disease, one that a person can get from something. It doesn't have to be something that is communicable disease, although in the future, I'm thinking about making it a requirement that it's a communicable disease. But at the moment, it's not. So you can pick any infectious disease, a disease caused by a microbe. There are some caveats. Like I said, you have to be the first to request it. And second, it has to be an infectious disease. Oh, what's the third? I'm losing my memory here for what the heck I was going to say. Oh, and that is, it has to be a single disease. So if somebody requests pneumonia, I'm going to say back, you can request one agent that causes pneumonia, but there are many bacterial, viral, and fungal agents that when they get in the lungs cause pneumonia. And I'm not going to let you do the assignment on pneumonia at large, covering all of those different topics. You have to pick one agent that causes pneumonia. Okay. And that would be another way we could break down, like someone done a pneumonia, there's streptococcal pneumonia, Klebsiella pneumonia. I'm sure there's viral agents that cause pneumonia as well. And then I think Candida albicans is a yeast that can cause pneumonia. And then there's another fungal disease that causes pneumonia. Okay. Any questions about that? Oh, good. The lecture Zoom files just ended. That means my computer will be working at full capacity. It won't be slowed down by the Zoom. It does something to the file so it can make it so that you can see the Zoom meeting. It makes it I think not a PDF file, what do you call it? MPG file or something like that. Any questions about the infectious disease project? If not, let's go ahead and talk about that. I'll talk about that before talking about lab 2 aseptic transfers. But that's what we're going to do today. Talk about the infectious disease project and about lab 2. Let me state that you do need to read textbook 3 through the section covering compound light microscopy. You need to read that before lab 3, which we'll have on this Thursday, and before lab 4 on stains, which will be next Tuesday. So you need to read that before beginning labs 3 and 4. Any question about any of that? If not, let me close down a few things here. And then start talking about the infectious disease project. I thought I logged into Canvas. That's another course. So let me get back into Canvas. I must have closed it down. And I really liked it when I just logged into Canvas, got on my class, and then there was a link for Zoom. And for some reason, they removed that link. So I have to get into Canvas the old-fashioned way, which I haven't done for years. Let me see. I think everybody took the plagiarism quiz. I checked that on Friday. And everybody submitted lab module 00 by Friday as well, which is nice. I didn't have to drop anyone. Remember to take the practice quiz 1B, July 19th. That's actually tomorrow, Wednesday, by 11:59 p.m. And here's the infectious disease research project. Actually, instead of doing it that way, I should just go to my hard drive and get it that way. So the infectious disease research project is going to be a project worth 100 points. It is a lab project, so the grade will go on your lab course, but it is worth 100 points. In this project, the goal is to make you an expert on one infectious disease. Any question about that? You can read about the rationale of this project. For the format, you need to pick an infectious disease and check with your instructor about the disease. If no one has your disease or has requested your disease, I'll get back to you with it and tell you that it's fine as long as it's an infectious disease. If it's not an infectious disease, I'll tell you it's not an infectious disease and to pick an infectious disease. And then if it's a disease which is caused by more than one organism, then I'll tell you to pick one organism that causes that disease and do your project mainly on that one organism that causes the disease. You may briefly, in just a few sentences, discuss the disease at greater length, but do not spend much time or effort or much space on your project, meaning your report, on the other agents that cause that disease. You'll need to be assigned that disease by me, and you will perform a search of the literature on that disease. And let me state that I've had a few students lately who will wait to the very end. And if you wait to the very end, you got to send me a large number of diseases, because most of the diseases will be picked. So you need to send a large number of diseases. And then I had one student who waited to the very end and said, well, I don't have time to get back for you, so I'm choosing to do it on this. And I told her, we know you didn't get your disease approved, and so there's going to be a penalty. So if you don't get your disease approved, there will be a penalty. Okay. And I didn't like that the fact she chose a disease that somebody also had chosen that disease. And I want each disease to be independent and different. So that's the reason why you have to clear it with me. So perform a search of the literature. You can search scientific books, magazines, news media articles that are good, scientific journals, and the internet to learn about your scientific disease. However, when you get this information, make sure it's a good source, because 4 of your formal full citations have to be a source which is good, and has to be a source which is peer reviewed. Your textbook is peer reviewed. Scientific journals are peer reviewed. I don't believe there's any scientific journal now that is not peer reviewed. The internet may or may not be peer reviewed. News media articles like Time magazine are not generally peer reviewed, meaning some person writes it, the editor will look at it, but will not check the facts of the article and then will publish it. Magazines, many magazine articles are peer reviewed. All textbooks are peer reviewed. And like I said, 4 of your full references have to be a peer reviewed reference. And generally I can just look at the reference and determine whether it's peer reviewed or not. I'm that good at just looking, and I can tell you whether it is or not. And it must be a good source. So make sure that if you're getting information from somewhere, it's a good source, like Time magazine, or is it Time, the newspaper? Yeah, the New York Times is a good source. The Oregonian is a fairly good source, but the National Gazette, Sun magazine, what are some of those other magazines at the grocery stores, which are not any facts that they publish? You can't use those for a reference, okay? And then other bad sources would be a source from a political organization, a personal website or personal site, a religious organization, because they will have a bias. You want the information to be factually correct. Any questions about that? All right. So write up your report. And when you're writing it up, please use a type that is single-spaced, 1-inch margins, and 12-point font. And if you notice, I wear glasses, and anything smaller than a 12-point font will be hard for me to read. The written report must include the following items. It must have a cover giving the title, the name of the disease, your name, the date, and the course name. I do not want a cover page. Just put the cover on top of your first paragraph of your report. I used to print these out, and if I printed it out and there's a cover page, that would be just a waste of an entire page of paper. Okay, it's just waste of space if you have a cover page in my mind, so I do not want a cover page. Just have the cover information and put it on the top of your text. The report should have the text, the main body information. The text should not exceed 5 single pages of text. If it's less than 5 or more than 6, and I guess I should say if it's 4 or less or more than 6, you will have a penalty, meaning I don't want it to be a large report and I don't want it to be a small report. Now, what I'm talking about is the body of the text. I'm not counting pictures. I'm not counting references for this. Any question on that? It must include all aspects of the disease organism that I'm asking you for, and that I'll talk about in the report content section below. The text will be worth 60 points, an additional 15 points for grammar, spelling, flow, and readability, meaning you will be graded on how well you write. So if you have poor grammar, poor flow, poor readability, you should get your paper done early and then take it into a writing lab at Clark College. Any questions about that? You should use as much as possible your own written words. You may use direct quotes only when absolutely necessary. So any information that you're getting from someone else, and that's ever 3 words or more, it should be put in quotes and say where you got it from. Okay? And I once had a student turn in 50% of her paper, which was the infectious disease paper, that was in direct quotes. And I didn't like that. So I will tell you that if you have anything over 20% more of your material in direct quotes, I will apply a penalty. And it won't be great unless you have like 50% of the paper in direct quotes. That's way too much direct quotation. Okay, it's not plagiarism because she did put it in quotes, but I do want it to be in your own words. If you do plagiarize someone, and this paper will go through a plagiarism check, you will get a score of 0 on this paper. And 0 out of 100 will really affect your grade. Generally, if the plagiarism is less than 20%, I will not look at it. If it's less than 30%, I will look at it and see where the plagiarism is being done and where it came from. And then I'll make a judgment there as to apply a penalty or not. Like if the plagiarism is coming from your references, will I expect your references to be plagiarized? Because everybody who turns in those reference will have a similar or it's the same reference. Okay, so that should be a plagiarism there. And then occasionally people, when they're just typing, they can type something similar. And that's why I will ignore any plagiarism that's less than 20%. If your plagiarism is more than 30%, you will get a penalty. Any question about any of that? And you can actually take a look at your plagiarism score after you submit the paper. The plagiarism check will be done as soon as you submit the paper, or at least a few seconds after you submit the paper. Any question about that? All right, you must have full text citation at the end of the paper, and you must have in-text citation inside your paper. Let me show you how to do an in-text citation. And I suppose while I'm doing it, a full text citation. So we have a bunch of text. And after your text, if you've stated a fact that isn't commonly known, you should have a in-text citation. Give the author's last name, and the year of publication. And you put that within parentheses. And it should just be the author's last name, comma, and then the year 2021. If ever it is, I'll give an example here. So that would be something like Smith, 2021. That is the in-text citation. If it's two people, you should have Smith and Jones, meaning both authors' last names, 2021, meaning the year. If it is three, and that J should be capitalized, by the way. If it is three or more authors, you go for an in-text citation, Smith et al. You know, I've forgotten, oops. It must be et al. I've forgotten how you do that, which is really bad. For some reason, my memory isn't doing too well today. Probably didn't sleep well last night. That's right, Smith et al. 2023. So you abbreviate whenever it's three or more authors. And this is about the only time you can use et al. In a citation. Any question about an in-text citation? Now let's break that down. In-text citation. The CCC just being the text. You have to use an in-text citation every time you give a fact which is not commonly known. Like if you say something like, a lot of people in the United States come down with the flu in a year. Well, that's a commonly known fact. You do not need to give an in-text citation. However, if you say something like in 2022, during the year, there were, I have no idea how many flu cases there were, so I'm just going to pick a number of out of the hand. 100,000. Well, let's go 1 million. 1, 2, 3, 4, 5, 6, 1 million flu cases in the USA, and there were, I don't know, let's say 100,000 hospitalized cases. So for that, you must have an in-text citation, because that's a direct fact, and very few people would know this. How many flu cases there were in the United States in 2022. I'm just pulling a number off the top of my head. I have no idea how many there were and how many cases were hospitalized, okay? So that should have an in-text citation. And you should have it, the in-text citation, after every fact that you state, which is not commonly known. Any question about that? There is one exception, and that is if you have an entire paragraph listing several facts on several different sentences. So let's just go 3 different facts, and they're all from the same reference. At the end of that paragraph, you may give the index citation. Well, I guess we shouldn't do a period there, because the period should be after the index citation. Any question about that? And that's only if all four of them, or whatever it was in the paragraph, all are using the same citation. If they're all using different citations, you must use the different citations, the index citations. Okay. You will get a penalty if you don't have enough index citations, and that means you're stating facts which are not commonly known. I've never given a penalty for having too many index citations, but if the paper becomes unreadable, you've got too many index citations. That is an index citation that happens inside your text of your paper. There's also a full text citation, which is usually just called your references. So I guess I should put that in parentheses because that's what it's usually called. Here, you may not abbreviate, and there is an exception, and I'm not going to discuss it. So for Smith 2021, if it's a journal, you give the author and you give at least his initials, if not the full name. So you could give like John Smith, and we're just going to go J.A. or J.S. 2021. Then you give the article's name. Journal name. Volume number. Page number. So if it's a scientific paper in a journal, that's how the reference is. Oh, I forgot something. Oh, I do. I got it there. Article name, journal name, volume number, page number. If, on the other hand, it's from a web page, you give the same start, the author, the year, the article name, that would be the web page name, and then the web page address. On this, you generally should give the exact web page address that you got the information from, but if there is a front page and it links to other pages, you may give the front page, assuming that one link is all you need to get to the reference that you're referring to. Is that clear? If it's more than one web page, then you should do something similar, which the article name will be too, because it'll be the different, it's not actually article name, it's the page name. Let me change that here. Page name too, and then web page address too. So if it isn't a single click from the main page, you're supposed to give the web page address. Now, if it's a book, it's a little more complicated. Let's see if I've got that information there. So it'll be the name of the author, the year, the article name, the book name, the editor of the book, the publisher of the book, and then the city of publication. Many textbooks now are printed in more than one location, and you only need to give the one city of publication. I always give the first city of publication. Is that everything? I think that's everything. Okay. Any question about anything? The computer can actually get you the full reference, and they will put in a bunch of stuff after this, which are usually a bunch of numbers. And I don't even remember what those numbers stand for, but it has to do with the reference for something. Anyone know what I'm talking about? I think that's on web pages too. And I do not know if it's on the thing. And that's fine. You can include those numbers if you want. You can remove the numbers if you want, because the numbers don't mean anything to me. So you may include those numbers. And like I said, I think it's the library website will actually make the reference for you, and you can use that reference. Any question about any of that? Everything that is in a in-text citation should have a full citation. Not everything that has a full citation has to be an in-text citation. Like you can get some information and you just never use it to specifically back up a fact, and so it won't be an in-text citation. So the full citation can not be an in-text, but all in-text citation should be in the full citations. I used to check that, but I'm gotten very lazy and I don't check that very much anymore. So as long as I don't notice it, I'll probably let it slide. But in theory, all of your index citation should have a full citation. Any question about citations? I went into this in great detail because students frequently get citations wrong, either the in-text citation or the full text citation. And then you're missing points on the project. One thing that students frequently get wrong is they will abbreviate the full citation. Generally speaking, you have to write out all of the names of all of the authors. The exception is only if a paper has more than 7 authors. Then you have to write out something like the first 7. You can put et al. Or ... and then you have to give the last author. Okay, that's the only abbreviation you can give. So if you abbreviate, you're going to get a penalty, because the full citation should generally not have abbreviations. Okay, any question about any of that? What's the other thing that people get wrong on the citations? You must give the full format of the full citation. There's something else that students get wrong on the full citation. I'm just forgetting what it is. It might be that 4 of the full citations have to be a peer-reviewed reference. I'm going to state that you can use Wikipedia. In theory, at school, you're not supposed to use Wikipedia because anyone can go into Wikipedia and then change the information on the page in Wikipedia. but as long as you have other information to back up what Wikipedia says, and generally Wikipedia gives references, you can just check the references to make sure that they're correct. And oftentimes students will use Wikipedia and then they will give the references that Wikipedia gives for their own references. And that's another thing you can do. But I won't accept Wikipedia as a peer-reviewed reference because it's not. And you can give other references which will be peer-reviewed, and you can get many of them from Wikipedia. Any question about any of that? If not, let's move on to the project. In-text citations are worth 10 points. You can use any citation format you want. Just be consistent. Always use the same format in the same paper. Sources you cite must be scientifically appropriate and reputable, like the USDA, FDA, CDC, scientific journals, popular magazines like Scientific American. A variety of scientific reputable television shows from like the History Channel, PBS, are acceptable. Religious, politically, and privately funded websites or personal websites are not acceptable. College websites where you contain student information are not acceptable. If you have a college website which is made by a professor, it is generally reliable. It's not reviewed, like my webpage is not reviewed by anyone, but if I find an error, I will correct it. So it is generally reliable. And I have not found an error on my webpage for many years. I say no Wikipedia, but if you want to give it, I'll allow it. As long as you have 4 references which are peer reviewed. And I thought I had that on here someplace. I'm not seeing it. Someplace in here it says that there must be 4 references which are peer reviewed. I may check up a full citation to make sure it's valid, especially if it's a web page. It's very easy to do. The full citation will be worth 15 points. Oh, there it is there. You need to have 4 unique sources referenced in your report. And by unique, I mean peer reviewed. You must submit the report as a document, a docx document. These are Microsoft Word, rich text format. And I think I've disallowed PDF. So let's remove that. The reason for no PDF is that the plagiarism check cannot check a PDF document. Other documents like, what do they call that? WordPerfect, I cannot open, so I will not allow you to add it. If you need to, and you have another word processor, then just save it in a rich text format, RTF format. Okay? It doesn't have to be a doc or a doc. I might even allow a text format. I have to look into that. Oh, another one is Write. Write is WordPad. So that's allowed as well. All right, the content of your document, your text should have subheadings, each discussing something. The subheadings will be the causative agent causing the disease, the history of the disease, the epidemiology, the pathology, the response and treatment, and the social, political, and economic considerations. You must have these subheadings or you'll get a penalty. You must then break down your report into these subheadings. So talk about the causative agent. Obviously, what you talk about will depend on what the causative agent is. If it's a bacterium, discuss the Gram reaction of the bacteria, the cell shape, whether it's a cocci or a bacilli or a spiral, the arrangement, the growth arrangement, metabolic capabilities, anything else that's unique to your agent causing disease. If it's a virus, you need to tell me whether it's a DNA virus, an RNA virus, whether it's a positive strand RNA virus or negative strand RNA virus, whether it's a retrovirus, whether the virus is envelope or naked. And we'll talk about these when we get to the lesson on viruses. You should talk about how large the virus is, whether there's a provirus, and is there anything unique about the characteristics of the virus. All right, this tells you about what to do for prions and eukaryotes. I'll let you read about that. And then move on. So in the next paragraph or about the next page, talk about the history of the disease. How long have we known about the disease? How was this disease treated historically? What is the history of the vaccine development? If there is a vaccine, if there is no vaccine for that disease, just state that, okay? And then describe the changes in our knowledge and attitude toward the disease throughout history. Even with COVID-19, a disease which is only a few years old, our knowledge and attitude towards this disease have changed, okay? And then an ancient disease, our knowledge and attitude towards those diseases have changed a lot. Like Mycobacterium leprosy or lepri, which causes leprosy, our knowledge and attitude have changed a lot. In the old days, in ancient times, like biblical times, people have to go around and say, I'm clean, to announce that they had leprosy, and that other people, if they wanted to prevent getting leprosy, they should stay away from them. And so they were sort of isolated from the rest of people. And even later, like I don't remember when, but maybe before 1950, yeah, before the development of antibiotics, even in the United States, we had a colony on, I think it was an unpopulated island in the Hawaiian Islands, where we sent the lepers. And that was to let them live together and prevent them from spreading the disease to other people, because that was before the development of antibiotics. And now we have the development of antibiotics. And so the lepers are living with us. And hopefully when they get leprosy, they go and get the treatment, which would just be administration of antibiotics. So talk about the history of disease, talk about the epidemiology, describe the prevalence and transmission of the disease, where in the world it's found, how many people are currently infected, what's the rate of new infections, what's the rate of morbidity, that's people who become sick, and then the rate of mortality, people who die of the disease. If nobody dies of the disease, just state that. Like the human cold, would be very few cases of mortality. And then state where the reservoirs of the pathogen come from and what are the modes of transmission of the disease. Talk about the pathology of the organism and the response and treatment. They should be in separate paragraph or pages. And then lastly, talk about the social, political, and economic considerations. I'm taking an awful long time, so I'm just telling you, you need to read about those areas when you write your report. I've got a copy of the rubric for the grading found on the next page. You'll get 1 point for your cover information, 9 points for your causative agent, 10 points for your history, 10 points for epidemiology, 10 points for pathology, 10 points for response and treatment, 10 points for social, political, and economic issues. And that will be the majority of your text. That's the 60 points. The quality of writing, 15 points. In-text citation, 10 points. Literature citation page, 15 points, for a total of 100 points. Any question about this project? All right. I'm going to shut that down. What did I save? Did I change something on this page? Let me go back and see.
I think you changed
Oh,
the
yeah, I changed
formats.
the format. I removed, I updated the format because I no longer take PDF files, and I just never noticed that until now. I've not taken PDF files for at least a year. So let me save that and close it down. We don't need that anymore. All right, so let's talk about Lab 2, aseptic transfers. I should mention I got Lab 00 graded. I'll try and get Lab 01 graded tonight, so that you'll have close to a week, about 5 days, to getting the what do you call it? The corrections put in, and then you'll get a second grading, which will be the grade that counts. So now, if you don't have 100%, you can make corrections and get it up to 100% of the lab. And I believe Lab 00 is worth 2.5 points. I'll always try and get the first grading done within a week of when you submit it. The second grading, I'm only going to get it graded when I have time, and I'm not going to have a priority on the second grading. The priority will always be on the first grading. So I'll probably fall behind in grading the second grading of each lab. Any questions about that? All right, if not, let's move on to Lab Module 2. Why do I have that in there twice? Oops, didn't catch it in time. Remember, when you turn in the lab, to only turn in the exercises for the lab, because it's much smaller than the lab module. If everyone were to turn in the lab module, my Canvas website would soon become full, and then nobody could submit an assignment. So please only turn in the exercises. The first time you turn in the lab module, you will get a warning. The second time you turn in the lab module, I will apply a penalty. Any question about that? If not, let's talk about Lab 2. Upon successful completion of this lab, you should, 1, be able to understand the purpose of using aseptic transfer techniques. You should, 2, be able to determine if aseptic transfer was used from the data provided. And 3, be able to define the following terms: ubiquitous, aseptic transfer, inoculation, and a contaminant. In our local environment, microorganisms are ubiquitous. This is a problem when working in the microbiology lab, because we generally want to work with a pure culture, a culture that only contains one species. If it contains more than one species, and then you run a test, you won't know which species caused the test results that you're seeing. It may be one species, several species, or maybe all of them, and you just don't know. So you have to work with a pure culture in order to understand the different tests. Louis Pasteur developed the concept of getting a pure culture, and I think I talked about that in the lecture, did I not? Nobody's answering. I think I talked about that in the lecture.
Yes, you did.
So I'm not going to talk about it here. He also developed the technique of making aseptic transfers to minimize the risk of contaminating your cultures, meaning accidentally inoculating unwanted organisms into a pure culture. We're going to learn how to make aseptic transfers in this lab. So obviously you should use tubes and cultures that are sterilized before making your aseptic transfer. During inoculation, never lay the cap down on the table or the lid of the Petri dish down on the table, because when you do, bacteria can be transferred from the table to the bottom of the cap or the bottom of the lid. And then when you put the cap over the container, something can fall down from the cap and contaminate the culture, or fall down from the lid and go into the culture. What you should do is, when you're working with a tube, you should hold the tube in one hand, hold the instrument that you're going to inoculate the tube with the other hand, and in the same hand, hold the lid to the test tube cap, like this. When you're working with a Petri dish, you should not totally cover the, you should not totally remove the cover or the lid of the plate. You should pull up the plate and then bring your instrument in, and then do your streaking or work on the plate where you're using the lid to partially cover the plate. That way, if something falls down from the air, most of the time it'll hit the lid. It will not go into the culture. It would have to fall down exactly right here to fall into the culture and then contaminate your culture. For the tube, it should be held at an angle. Maybe I can demonstrate this here. If you can see this cup, it should not be held straight up and down, because anything falling down will fall into the open tube. What you should do is hold the tube at an angle, and that way, if something falls down, it may fall on the outside of the tube and not contaminate the tube. It would have to be in this small area here to fall into the tube while it's open. And that small area is much smaller than if you have the tube totally open. So always hold the tube at an angle and then hold the lid on top of the plate while you have the plate open. Another thing you should do is, while you have the culture open, meaning the lid or the cap off, you should quickly get your work done. You don't want to leave the tube open for any length of time, or the plate open for any length of time. You want to have it open for as little time as possible, and that will reduce airborne contamination. So work efficiently, don't talk. While you talk, something can come out of your mouth and then contaminate the cultures. And then, of course, before you begin work, you should always sterilize the area, disinfect the area where you're going to be working. And then when you're all done at the day, you should disinfect that area to make sure anything that you dropped on the table will be removed or killed. All right, So in making aseptic cultures, we need to inoculate, that means to transfer cells to a new sterile media. You will get the sterile media in the front of class if you were to actually come into a microbiology lab, and it would be sterile because it had been put in the autoclave, and then it'll be cooled and then made available to you. You must make sure that we sterilize everything that can potentially contaminate the sterile media, meaning you need to sterilize the instruments you're going to inoculate the tube with. And you sterilize them either in a Bunsen burner or in a Bacto incinerator. Here is one of the tools we use, and we call this tool a loop. You'll note that it has a wire portion that is shaped in the shape of a loop, and then a metal handle portion, and then a plastic or rubber portion of the handle. And that's to prevent the heat from transferring to your hand. When we heat the loop, you only heat up the wire portion of the loop. You do not heat the metal portion, and you certainly don't heat the plastic or the rubber portion. How you heat it is you put it in the flame or the Bacto incinerator until all of the wire becomes red hot. And then you bring it out, cool it, and then you use it to transfer cells from like one tube, for example, into a tube containing sterile media. So this one has the bacteria in it. All right, know the terms of this lab. In laboratory 2, you're not physically going to be performing any of these activities, as all of our lab work will be done online, but you will be watching videos of these activities, and we expect you to know and understand the material in the videos and in this lab. Procedure 1 is going to transfer sterile liquid using a glass serological pipette. Procedure 2 is going to transfer a sterile paper disc using forceps into a tube. We have a little video to watch on demonstrating proper aseptic transfer techniques. So click that and watch it for the lab. In procedure 1, you're going to transfer 1 ml of sterile water into a tube of nutrient broth, and you're going to do this using a pipette. Now, when you're using a pipette or a Biuret, you'll note that liquid water does not go across the glass straight. Let me blow that up. Meaning the liquid water forms a meniscus. It's not straight across the glass. And where do you read the meniscus? Do you read it at the top of the meniscus, the average of the meniscus, or the bottom of the meniscus? Does anybody know?
Bottom.
Yeah, you read it at the bottom of the meniscus. Thank you for giving an answer. You should never mouth pipette in microbiology. You should always use a pipette pump, meaning something to actually do the pumping for you. Let's move down here to procedure 1. You need to use this table for interpretation of your results. So you should be familiar with these possible outcomes and the associated terminology. For example, you will be asked to state what the results are for procedure 1 and the conclusions of that result. So in procedure 1, if the nutrient broth is clear, then no organisms are growing in the broth. And so you should state that no organisms are growing in the broth, and aseptic transfer was successful. So you must use the wording of this table to describe the questions in the lab that ask you about the results and the conclusions for procedure 1. On the other hand, if the nutrient broth is turbid, meaning hazy or cloudy, organisms are growing in the broth and aseptic transfer was unsuccessful. Any question about that? So come down here, and here we see for procedure 1, the tube as it is shown before you inoculate it. And as you can see, it's clear. There's no growth in it, and it's sterile because it was put in the autoclave. And then come down here to procedure 1 to see the results. We're supplying the results in pictures because you can't do this obviously online. P1 stands for procedure 1. So this tube is the results of procedure 1. Remember, we inoculated a tube of broth with 1 ml of water. And then answer the questions. Describe the initial clarity of the nutrient broth prior to inoculating, meaning prior to adding the sterile water or the paper disc. And I think you guys will have that as question 0. So you need to state whether that was clear or turbid. And then for 1, describe the clarity of the nutrient broth after inoculating with sterile water and overnight incubation at 35 degrees Celsius. Was it clear or turbid? Based on these results, what conclusion can be drawn? And let me emphasize for the last time that your wording must be similar to the results and the conclusion of the table here. So you don't need to give the results, but you need to give the conclusion. And then list 3 sources of error with the aseptic transfer technique that could explain why the nutrient broth showed the growth of microbes. Okay, so this will be 1A, this is 1B, this will be 1C. And sometime in the future, I'll actually label that, but at the moment they're not labeled. And so this will be 1A, 1B, and 1C, and it's pretty obvious, at least to me. I've never had students having a trouble with that. For 2, well, let's go back to procedure 2, coming up here. Procedure 2, you're transferring a sterile paper disc into, sorry, my mouse is not working. Let me go this way and then go forward. There we go. That worked. Procedure 2, you're using aseptic transfer to transfer a sterile paper disc into the nutrient broth. And the picture showed you what the broth looked at the start. And then we're going to show you, the procedure is given here, what the results are for procedure 2. In the picture down here, here is procedure 2, tube P2. And there you can actually see the paper disc that was transferred into the broth. So give your results. Is it clear or turbid? And then based on the results, what conclusion can be drawn? The wording must be the same or similar to the table. And then that's it for procedure 2. For procedure 3, coming up here, we're using aseptic technique to transfer Serratia marcescens into sterile broth. Now, broth cultures are going to be problematic because if there's minor contamination in the tube, the contamination will mix with the culture growing in the tube, and you will not be able to distinguish minor contamination from the real bacteria. Okay. However, if there's major contamination, you could possibly detected. And the reason why is Serratia marcescens is pink in color when it grows in a broth. And so if there's major contamination by something that was white or yellow, it won't be this pink color. I guess white would be a very light pink, but yellow would be like an orange color. You must use the table to explain your results. For procedure 3, if the nutrient broth is clear, no organisms are growing in the broth, and aseptic transfer was unsuccessful. Can anyone explain why aseptic transfer was unsuccessful if we had nothing growing in the broth?
Because we wanted to transfer the bacteria.
Yeah, because we want to transfer Serratia marcescens into the broth, so we're expecting organisms to grow in the broth in procedure 3. So if the nutrient broth is turbid, hazy, or cloudy, organisms are growing in the broth, and that's what we expect. So we can assume aseptic transfer was successful. Let's look at the results. P3 to P3 stands for procedure 3, and you can see the results here. Let's go to the questions. P3A, describe the clarity of the nutrient broth after inoculating with Serratia marcescens and overnight incubation. Is it clear or turbid? And then based on these results, what conclusion can be drawn? So that would be 3A and then 3B. And then 3C, how do you know whether you transferred the correct microbe, Serratia marcescens? And I'll give you a hint. Serratia marcescens is pigmented, so you can know if you have the correct microbe by the color of the tube. Okay, results 4 is transferring Serratia marcescens into an agar slant. The results for procedure 4, if the slant does not show growth, then no organisms are growing on the slant and aseptic transfer was unsuccessful. If the slant does show growth, then organisms are growing on the slant and aseptic transfer was successful. Here we see tube P4 for procedure 4. You can see that when Serratia marcescens grows on a slant, it's actually reddish in color. It's not pinkish, it's reddish. And then for the questions, is there bacterial growth on the slant? You just need to answer yes or no. How do you know? So state how you know, yes or no. And then for 4C, does it appear to be the bacterium you inoculated or a contaminant? And you should give a little bit of a reason for why it's your answer is the thing. And then that's it for lab 2. Okay, remember to answer your questions on the lab exercises, which I never showed you. Let me go to there now. It's the same questions in the lab module. And then submit the lab exercises for this lab. And submit it by Saturday at 11:59 p.m. And then I will grade it for the first grading. Any questions about this lab? If not, go ahead and get started with the lab. You have some videos to watch on procedures, and then obviously read through the lab and then answer the questions. I will be here until 9 o'clock or until the last student logs out to answer any questions. I do make myself available to you because, well, you're my students and I want to be available to you to help you. It is my goal in life to help you. As far as I know, I'm the only person who sits in a lab and will wait for students to answer questions. All right, get started in the lab, and I will be here until 9 o'clock or until the last student logs out. I guess I should ask, are there any questions before we begin?